Okay, welcome everybody. It's a great pleasure to introduce today's speaker, Markus Bastir. Markus actually is Austrian and also did his master's project with Horst Seidler at the University of Vienna in 1998. Then Markus went to Madrid to the Museo Nacional de Ciencias Naturales, did his PhD there with Antonio Rosas, finished in 2004. Thereafter, he spent 2 years at the University of York with Paulo Higgins. Then he went back to Madrid as a Marie Curie Fellow and later as a Ramón y Cajal Fellow. Since 2010, he's a senior scientist at the Museo Nacional de Ciencias Naturales in Madrid, and now he's a research professor there. Markus also is a research associate at the University of Witwatersrand in South Africa. And today Markus is here. Markus actually is spending time in Vienna now in the course of a sabbatical, and we'll talk about the building blocks of the human body. Please, Markus.
Thank you very much for the nice introduction. Thank you everybody for coming here and giving me the opportunity to talk about the research we are carrying out in our lab. And if I manage to switch here, yes. I want to talk about our work on the origin of modern human body shapes and how the trunk anatomy and function contributes to this. It is generally known that modern human body structure is characterized by a tall body shape with elongated inferior limbs, a tall and narrow trunk. And from the craniofacial perspective, modern humans are characterized by a reduced face and a large brain case. And from the postcranial part of view, which concerns this lower part of the skeleton, it is actually thought that this body structure appeared some 2 million years ago. You can see how this differs to other representations of body reconstructions, and the adaptive background of the appearance of this body shape is related to a paleoecological change from tropical forests, which was the preferred habitat of Australopithecus, to open grassland and dry hot savanna, which is supposed to be the new habitat of the genus Homo. And you can see here that these landscapes also somehow demand different body adaptations. Australopithecus is adapted to arboreal locomotion as well as to facultative bipedalism. And in the context of these locomotion patterns, the few skeletons that are reconstructed suggest that the upper part of the thorax is relatively narrow, which provides a mobility of the upper limbs, and the wide everted pelvis somehow holds the necessary size of the intestines in relation to plant diet. And the longer limbs are related also to this arboreal locomotion. In turn, the first representation of early Homo and complete representation, which is usually referred to the anatomy of the skeleton, which is formally known of KNMWT 15,000 or 15K, but informally known as the Nariokotome boy. This is characterized by already these elongated lower limbs, and they are thought that these are kind of adaptations to an extended ranging behavior which became necessary in this kind of different landscapes. And what is interesting is that body shape is actually relatively straightforward to measure in recent humans, as you can see on the left side of the slide, but it is much more challenging to be assessed in fossil hominins. This is usually carried out by relating some kind of long bone measurement, which could be an estimate of the stature, and some kind of pelvic measurement, which could be an estimate of the width. And so body shape is basically characterized by relationship of heights relative to widths. But in the case of the Nariokotome skeleton, we have a problem because there is no pelvis. The pelvis, well, there are some remains, but it needs to be reconstructed. And one of the big problems with this pelvis is that there is no true anatomical element preserved that would connect the right and the left part of the pelvis. So we have no idea about the mediolateral variations. We do, however, know that the long bones are somewhat indistinguishable from modern humans. And so the question is, how can we reconstruct those measurements that we want to have, that we need in order to get some idea about body shape? And if you can see on the left side, it's possible to put all these fragments together and come up with a relatively narrow pelvis, but it's also possible to reconstruct them in a way that gives rise to a more heavily built and wider pelvis. And so actually two different kinds of phylogenetic models about the origin of modern human body shape are possible. And therefore it's interesting to see if there are alternatives that can be used to answer this kind of dichotomic questions. And this is important because one of the crucial factors that are assumed to be involved in the change of body structure are related to a specific subsistence pattern, which is known as endurance hunting. There are several images here who show the kind of landscape, and one of the most classical ones is here, this picture of the Bushman running through the savannah and chasing quadrupedal game. And the idea of this endurance hunting is to use endurance running, long distance running, and to pursue the animal and to run it into a hypothermia. And this would be a kind of long distance running on low speeds, metabolically speaking, on aerobic metabolism. And eventually the animal gets into hypothermia, and then it's more easily being killed by the hunter. And this model was actually put forward already in the 1980s by David Carrier, and then it was more refined by Bramble and Lieberman and co-workers. And the interesting thing here is that if we look at the velocities of running in humans, then we can see that the human endurance running speed shows a wider range compared to quadrupeds. And the interesting thing is also that actually all of the quadrupeds are somehow inside the endurance running velocity range of the modern humans. And therefore, it is thought that the cost of transport in human is actually not very variable. And this is related to the fact that in quadrupeds, the energy of the locomotor system, the respiratory system, and the thermoregulatory system, they are integrated. They work as one unit. Whereas in bipedalism, due to the specific body structure, the locomotor system is separated from the respiratory system and independent from the thermoregulatory system. These relationships can be somehow imagined as if you run, it depends on your running speeds. You can modify your speed of breathing. And because we have a thermoregulatory system that is not depending on panting, but rather on sweating, we don't really need our breathing in order to cool our body temperature. And so it's interesting to know that specific adaptations of the human body, particularly with respect to the trunk are interpreted of these endurance running adaptations. And those are mainly a narrow pelvis. Here you can see the comparisons of Homo erectus and modern humans in comparison to Australopithecus. A narrow lower thorax, which we have seen already, is a key feature between modern humans and if we compare it compared to the Australopithecus, and a tall and narrow waist. And all these features are important in the context of running, and particularly in the context of energy saving during counter rotations of the trunk versus the hips. And it is thought that it is Homo erectus where these features appear first in evolution. And so it makes actually sense to ask how narrow or how wide was the trunk of early Homo in order to fit the model into the paleoecological framework. And in order to answer these questions, we somehow introduced the concept of building blocks and wanted to focus on the evolution of the body plan, not in the context of a kind of monoblock, but we divided it into three parts, which is the thorax, the lumbar spine, and the pelvis. And we're interested whether dividing the trunk into these models can somehow provide a better answer to this question of origin of body shape. And so that's the work that was done in my former project in a PhD thesis of Nicole Torres Tamayo, where we took the old models of Adolf Schulz. You can see here the drawings where the width of the thorax and the width of the pelvis appeared somehow correlated. And we tested this with a quantitative model, so used different CT data from African and European populations. Nicole did a tremendous job to measure all these torso skeletons with 1,030 landmarks in 120 torsos, and we correlated the data from the pelvis with the data from the thorax. And so what we actually found was not surprising, but we could find a quantitative description of this pattern, that the width of the thorax and the depth of the rib cage are totally correlated with the width and the depth of the pelvis. And therefore, we had the possibility to, if we do not have a correctly reconstructed pelvis, to estimate body shape from the thorax. But that means that we have to build together the thorax. Here you can see on the one hand, the already known fragmented pelvis, but the CT scans of the thorax show that this is fairly well preserved, except for some missing elements and minor breakages. But we wanted to develop a method for a standardized reconstruction and searched therefore for methods that could provide a quantitative and reproducible reconstruction, because fitting all these parts together needs to be based on some comparative work. And so we devised first a method to estimate metameric elements. And so we measured a large database on vertebrae of the entire spine and established trajectories that describe the serial morphological shape change between cervical, thoracic, and lumbar vertebrae, and came up with some methods that were able to estimate the missing morphology of neighboring preserved vertebrae. And so the method consisted of first estimating the shapes with geometric morphometrics and then warping the 3D meshes in order to fit them into this system. And here you can see work from Carlos, who is focusing on thoracic and cervical vertebrae, and work from Dani, who worked more on the lower part of the spine. And we actually were successful and could reconstruct all these elements. Here you can see the brownish photorealistic models are the original surface scans of the fossil, and the gray models are the virtually estimated meshes. And we could use these methods not only for reconstructing missing serial elements of the spine, but also of the ribs. And so at the end, we came up with a complete set of ribs and vertebrae of the thorax that could be compared in a comparative ontogenetic setting. And what we found was that the vertebrae of the Nariokotome skeleton were characterized by a dramatically reduced height of the vertebral body, a considerable extension in the anterior-posterior direction due to the orientation of the spinous processes, and by a backwards orientation of the transverse processes of those elements that articulate with the ribs. And on the other hand, what we see in the ribs is that the ribs of Homo erectus barely show some evidence of torsion. Here you can see in the modern humans that the sternal ends are much lower than the vertebral ends. This is not the case here. And at the second level of comparison, we can see that the cranial ends are lifted upwards relative to Homo erectus, who shows this plain, this flat morphology. And so we started developing methods for a standardized rearticulation of all these isolated elements. And this is also part of work of Jose, who is in his PhD is working on reconstruction methods of this musculoskeletal system of the rib cage. And we did both. We did first reconstruction on the spine alone. This is a kind of puzzle. So we got CT scans of patients, disarticulated by segmentation, the entire spine. Then we commingled everything, and then we put it together. The only thing that we did, the advantage that we used here in this method is we didn't separate the landmarks from the bones, so we had no measurement error. We just really could assess the original morphology and the different reconstructions. Actually, this was a project carried out by different researchers on different days. And so what we found is that if we articulate according to the posterior articulation of the zygapophysis, then we can really find a reconstruction where all of the reconstructions are closer to the original than to other spines. And we did something similar to show how the ribs can be fit to the spine. And this is just recently published. That method shows that if we do this along several days, then there is a learning curve, and there's actually very little difference from the original and the reconstructed one. And if we do that, we have a kind of before-after comparison. We come up with this kind of morphology of the trunk of the Homo erectus skeleton, where particularly the thorax is reconstructed. This is the old pelvis, just as a side note. And this is the morphology. If we now compare that in kind of standard measurements to the modern humans, then what we can see here is that in the upper part, it's actually the width and the breadth is an upscaled version of the ontogenetic trajectory of the modern humans. But if we combine it with the height, then we see that it's considerably shorter. Like the spine is much shorter in Homo erectus than in the modern Homo sapiens. And this is what I tried to summarize in these building blocks here. Now, also from the geometric morphometric point of view, we find clear evidence that the morphology of this Homo erectus reconstruction is outside the range of the modern humans, and that allowed us to reject the model of the deep origin of modern human body shape and rather proposed a recent roots model. And now the question is, what does this different morphology actually mean in a functional context? What is a deep and wide and short thorax meaning for running and respiratory function? And we focus basically on the interaction between the locomotor system and the respiratory system, because thermoregulation is quite well studied, but locomotor and respiratory system is connected to each other. we see that lung ventilation is on the oxygen which come into the circulatory system, and it is there used to distribute the energy to the cells. But it is also known that because the skeletal muscles that are moving the rib cage, we distinguish here between inspiratory and expiratory muscles, and it's also important to distinguish between normal breathing and exercise breathing. Because in rest breathing, expiration is mainly passive and inspiration is mostly brought about by the diaphragm, but during exercise, a lot of other skeletal muscles come into play, and they require energy. So the biomechanics of the breathing is interesting to know. And here's just a quick summary of those muscles that act on the rib cage. For the inspiration, we have the sternocleidoid and the scalenus muscles, as well as the external intercostals and the diaphragm. And for the expiratory muscles, we have mainly the abdominal muscles that tear the lower part of the rib cage and produce a forced expiration. And so if we now connect again the respiratory system with the locomotor system, it has been proposed by Dominelli that during heavy exercise, both systems compete with each other for energy. And therefore it makes sense to understand what shape variation in the context of breathing kinematics could imply in an energetic context. And therefore, in the second half of my talk, I just want to show some different experimental approaches that we are carrying out in order to address this question. And first of all, we want to show you some experiments on how do we use geometric morphometics to analyze breathing motion per se. This is motion recorded in a computer tomographic scanner. We worked with a set of volunteers. Here you can see myself and Dani participating already some years ago, and our rib cages in red in inspiration and in blue in expiration. But we did this also for a greater sample, and what we found was that the shape changes between in and ex, inspiration is the upper part, expiration is the lower part, are characterized by a kind of different relationship of the upper parts relative to the lower part. Because for the same kind of shape changes in the upper thorax, there is much less size differential produced due to breathing kinematics than in the lower part, which means that if the lower part is a little bit wider, then there is much greater ventilatory volume that can be moved in breathing motion. And so this is potentially interesting in the context of our fossil reconstruction, where we have seen that the lower parts of the Homo erectus ribcage is wider than the lower part of the ribcage in modern humans. And so we made a kind of forward simulation of the motion of the ribs within the articulation of the thoracic spine. And it turns out that due to the anatomy of the ribs, the complex 3D curvature, the inspiratory motion in Homo erectus, which you can see here on the left side, produces less superinferior motion and much mediolateral expansion than in the modern humans. And because we analyzed this motion study with landmarks, we can represent these shape changes here in this PCA, where you can see that is the modern human that changes mainly in a superinferior direction, whereas the Homo erectus reconstruction has this kind of mediolateral component in the inspiration. And so if we calculate also the sizes of these differences, then you can see that this is the range of the modern human size, and this is the functional size produced by inspiration in the Homo erectus. So the lower part really is capable to produce a much greater tidal volume than in the modern human rib cage. And this is certainly also compatible with work that HEAS is doing in his PhD, where he tries to reconstruct the soft tissues of the muscles. So we can see that also the diaphragmatic reconstructions is shows a much greater surface area than those of the modern humans. And so in the next step, we started comparing ribcage morphology just in sprinters and endurance runners to see if this kind of activity has some impact on the respiratory system function. And here you can see part of the team, but we were happy enough to be able to scan 20 sprinters and 20 endurance runners in a more or less balanced relationship between males and females to account for sexual dimorphism. And what we found was that there is a systemic difference between sprinters and endurance runners in a way that the sprinters show some narrower lower parts of the rib cages and slightly flatter ones than the endurance runners. And if we start, this is actually work that's in progress currently, but our interpretations for the moment suggest that the biomechanics of this rib cage, particularly in the lateral view, suggest that there is a longer lever arm for the abdominal muscles to pull the rib cage down in a forced expiration if we compare that to the sprinters. And so we think that there is the capacity of a powerful expiration in the long distance runner. And again, the greater width in the lower part of the endurance runner rib cages compared to the sprinters, following this similar principle, would suggest first, greater diaphragmatic muscle length, which means a stronger capacity for respiration from the diaphragmatic point of view, but on the other hand, a greater capacity of expansion and of course, more intake of air and maybe also some relationship to the diffusion capacity of the oxygen. And the interesting thing is that if we compare now the rib cage of Homo erectus with those of the runners, what we see is that we have this kind of different orientation of the ribs, which means that we have these better expiratory lever arms, but on the other hand, we have a reduced spine, and the shorter part of the spine actually causes that the Homo erectus torsoskeleton barely shows a waist. Like there is a reduced abdominal space in this vertical perspective, and this is work on the simulation of the respiratory biomechanics that shows that this space is needed in forced expiration. And if it is if this reconstruction shows that there is limited space, this means that there is no great capacity of powerful expiration such as we find it in the modern humans. And so in the next step of experiments, we just looked at how torso shape relates to running performance and running velocity. And there we analyzed a set or a group of volunteers that were both endurance and power locomotion specialized people, like runners on the one hand and rugby and soccer players on the others. And they were subjected to a stress test, which means that they were running on a treadmill, and we calculated the percentages of the heart rate reserve to determine 4 different categories of running intensities. And this is important because it is known that around 85% of predicted heart rate reserve, there is this kind of transition from the aerobic to the anaerobic threshold. And this is interesting in the context of long distance running. We also analyzed the shape of the torso, which means that we scanned the athletes after the experiment. And Jose Maria carried out his PhD thesis to develop the template for torso shape quantification. And here we used two different landmarks. We used one set that just quantified the entire shape of the torso, and another more reduced one, which focused more on those parts of the torso that were not covered by the muscles, to have some clearer relationship to what could be the skeletal structure of the thorax. And then we used regression models to relate the shape of these athletes to the velocity and to control for stature and body weight. And what we found was that actually there was no relationship between body shape and low running velocity. Only in higher speed ranges, we found a significant relationship suggesting that narrower and flatter people had higher running velocities than wider and deeper configurations of chest walls. And interestingly, if we compare now these running velocities to those proposed by Bramble and Lieberman, what we find is that actually the average jogging speed, according to this paper, is around 12 kilometers per hour. But faster people can run until 20 kilometers per hour, which is the speed needed to outrun the horse in a kind of long distance race. But what we found is that only in the step from 10 to 14 kilometers per hour, the relationship between torso shape and running velocity emerged as statistically significant, which means that we are not quite sure if the anaerobic transition actually occurs here, because there are many factors that determine this transition. But if we are on the lower part, then we would have no relationship between running. But if we are on the higher part of the supposed endurance range, then we do have this kind of relationship between torso shape and running speed. And the interesting thing is that what's similar between the running experiment and just the CT scanning comparison of sprinters and long distance runners is that in both cases, the high velocity people are characterized by narrow and flat lower torso morphologies. And this somehow relates to the proposition of Lieberman and Bramble and Lieberman in their review paper. But what now happens to the waist? Because it seems that there is some indication in the lower part of the rib cage. And in order to understand what's going on in the waist, which is something that is actually not conserved in the fossil record because it's only soft tissue, we carried out some other experiments where we work on the quantification of the shape of the chest wall during exercise breathing. And this is work that we carry out in collaboration with engineers from the Politecnico Milan, Antonella Lomauro and Andrea Aliberti, and it is the PhD work of Marta in our group. And she tries to somehow translate a very well-established way of measuring lung function, which is optoelectronic plethysmography, into a geometric morphometric protocol of motion quantification. And here you can see these experiments. People carry a set of 89 infrared markers. They are surrounded by a set of infrared cameras that record with a frequency of 100 hertz a lot of landmark configuration during breathing cycles. The volunteers undergo, again, a stress test, a so-called steep ramp test, which means that the resistance is increased until they are exhausted. Then they stand up and breathe in a standing position. And this is what is then recorded by the cameras and is what we analyze in our experiments. We used different moments in the breathing cycles So we use the volume to divide the cycle into 4 different positions: full expiration, half time between X and in, full inspiration, half time between inspiration and the next expiratory cycle, and analyzed this kind of data with geometric morphometric toolkits. And the first thing that we saw is that we have a kind of separation between flat and narrow chest wall configurations versus deeper and wider torso shapes. And if we remove the variation due to the individual shape, we only keep that kind of variance that is related to motion. And what we find here is that the deeper, which can somehow be thought of the increased depth that we found in Homo erectus, they use a different muscle activity pattern for forced and for exercise breathing than the flat ones. The deep ones seem to work mainly with the diaphragm and the abdomen, whereas the flat configuration also uses thorax in addition to the diaphragm and the abdomen. And this is interesting because if we now compare these breathing patterns of the deep versus the flat, and we consider that this is a flat configuration and this is a deep configuration, all the abdominal muscle action carried out in the simulations, in the analysis of our experiments, would somehow be affected by the absence of the waist. So we think that these comparisons somehow shed light on the fact that the waist is not only important for the biomechanical stuff in the context of running, but it is also very important in the context of exercise breathing. And in the Homo erectus reconstruction, as it is here, there would be just little space for this kind of recruitment, of volumetric recruitment during exercise breathing. And so I want to conclude suggesting that the evolution of modern human body structure may have occurred in 2 steps. The first steps is that the lower limbs appeared with Homo erectus, the elongated ones, and this is shared between Homo sapiens and Homo erectus. But a second step is that within the modern humans, only the tall trunk appears. This is a derived feature of modern humans, not present in the erectus, but it means from a paleoecological point of view, probably that Homo erectus was capable of endurance running, but mainly on a very low running intensity. And this is somehow compatible with the model. But the capacity of forced respiratory inspiration is linked to the morphology of the modern human trunk, and this is likely derived. We think it is related to a better exercise capacity and may have some implication in running with spurts or definitively showing a more intense running adaptations than the context of Homo erectus. And I want to thank you and all these collaborators, and particularly the volunteers, because this is really hard work. They are doing a lot of exercise, and thank you for your attention.